welcome back. Remember that these few seconds give you a little bit of time for your brain to settle in and get ready to learn. Okay, moving deeper into our discussion of DNA, and be talking about how DNA is packaged into our cells. How do we fit those 3.2 billion base pairs of genetic material into each and every single one of our cells? For this video, things to be mindful of thinking about is chromatin versus chromosomes. Okay, what are they and when is DNA in each form? Then types of chromatin and what characteristics of DNA causes it to be packaged in the two types. And then DNA representation, so karyotypes. So we're going to start to talk about why DNA is represented. So really chromosomes. Why are chromosomes represented different ways in different circumstances? Keep an eye out for those questions. So as I mentioned before, there are 3.2 billion base pairs in the human genome. That's a whole heck of a lot of A's and C's and T's and G's. A number that big is difficult to wrap our minds around. So just to kind of give you an idea, maybe kind of help, if you decided you were going to take a trip of 3 billion steps, you would go around the equator almost 46 times. If we were to take all of this DNA and stretch it all out end to end, all of the DNA in one of your cells, that would be about the height of your professor, about my height, with heels, but that's okay, I like heels. That's about 6 feet of DNA. That's crazy. I know as you're taking many classes and focused on learning, and there's so much stress, and I get that. But I hope every once in a while you can take a moment and just go, that's crazy. 6 feet of DNA if it was all stretched out in every single one of your cells. Okay? So we have to fit 6 feet of DNA in a cell, not just in a cell. That's not 6 feet of DNA across your entire body. That's 6 feet of DNA in every single one of your 30 trillion cells. And when we get through haploid diploid, which is coming next, I'm going to blow your mind again. So how? Probably pretty familiar with this. The fact that DNA isn't all stretched out and exposed so that we can read all of its A's and C's and T's and G's. Okay, we know that. It's not even just hanging out in its nice coil. But something that may surprise you is neither is it hanging out as chromosomes the vast majority of the time. Most of the time, DNA is in a form called chromatin. So let's talk about that. So if we take our DNA, this is one of our images that I talked about. We might see DNA all different ways. So if we take our DNA, we know that it's not laid out flat like this. We know that it's a double helix. The double helix structure is like the classic. It means DNA. And so think about, you have it, if you had it stretched out, if you had a piece of wire all stretched out and laid out flat, then if you took that wire and you coiled it into a spring, it's going to get a lot shorter. So this is the first step of condensing our DNA. And this is, the DNA coils like this just by the nature of the chemical bonds like we talked about. We talked about the phosphodiester bonds and hydrogen bonds. the particular way that the components of DNA bond causes it to coil this way, causes it to coil as a double helix. So then we take that double helix and we wrap it around proteins. So we take that double helix, we wrap it around proteins, and these particular proteins, they're going to be really important when we talk about transcriptional control. These particular proteins are called histones, and I'm going to just show you an image or several images here. And this is really the same concept that we talked about with DNA, that you might see these depicted in lots of different ways. So these are histones, histone proteins. These are groups of proteins that DNA is wrapped around. And so you might see them depicted something like this. You might see them like this, where you can really see those tails. Those are going to be important later when we're talking about transcriptional control. So the way that these are depicted depends on kind of what we're trying to emphasize, what information we're trying to give. So just keep that in mind, and also keep in mind with all of this stuff, when we're trying to create diagrams of DNA, when we're trying to explain these structures, what we're talking about is wildly complex molecular structures. So proteins are not just little circles that look like yo-yos. They're these incredibly complex, coiled and folded three-dimensional structures. But in order to talk about the components that we want to talk about, we sort of simplify them. So just keep that in mind. Okay, so going back, oh, I forgot that I animated that. Isn't that cool? Look at our histone. That's an animation of an actual histone protein. And I put the link at the bottom there for you if you want to go look at that. So our histones, the DNA wrapped around a histone together. So DNA plus histone equals a nucleosome. So those together are a nucleosome. So then DNA is wrapped around histones to form nucleosomes, and chains of nucleosomes then are called chromatosomes, and they kind of look like beads on a string. And you may notice up at the top in yellow that it says active genes. So genes that are being frequently transcribed, so the cell is using their products often, are considered more active genes. So active genes are coiled in this structure, in this way. Less active genes, those chromatosomes, are then taken and coiled on top of each other in this very tight structure here called a solenoid. And then those solenoids are kind of wrapped like this. So that's less active genes. So active genes, they are coiled, but they're kind of coiled more loosely, whereas less active genes are coiled very, very tightly. In both of these cases, this is how DNA is packaged during interphase. I'm going to show you more visuals of that in a moment. So both of these types, the chromatosomes and the solenoid, the active genes, the less active genes, this is all chromatin. So the cell has, every cell has the DNA packaged into chromatin during normal cellular activity. The different types of chromatin, so this loosely packed chromatin is called euchromatin. The tightly packed chromatin is called heterochromatin. Only during cell division is your DNA actually tightly packed into the structure of a chromosome. So just to kind of remind you about the cell cycle, so this chromosome, this incredibly, incredibly tightly packed DNA, so even more tightly packed than a solenoid, only happens during mitosis or M phase of the cell cycle. The whole rest of the time, all the rest of the time, when your cell is doing its business, carrying out normal cellular activity, living its life, living your life, your DNA is in the form of chromatin. And to give you kind of a visual, that would look kind of more like this. Okay? Parts of it are euchromatin, parts of it are heterochromatin, but it's all sort of this spaghetti looking mess of DNA. So on the next slide, we're gonna watch a video to kind of help us visualize that.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So if our DNA is so infrequently actually packaged as chromosomes, then why do we talk about our DNA that way? A couple of different reasons. If we look at this image, the image in the center, this is a cell undergoing cell division. Whereas the other cell that you can see pretty clearly in the image, which I'm going to circle here in green, is a cell that is normal cellular activity. So during that interphase, that whole period of time when the cell is doing normal stuff and the DNA is in chromatin. So you can see just from this image that we can't really tell much of anything about distinct DNA molecules in the cell that's undergoing normal cellular activity. Whereas in the cell that's undergoing cell division, we can kind of start to get some information. So that big long strand of DNA, like I told you, that 58 million base pair long strand of DNA that has 2,000 something genes, that's actually your chromosome number 19. And you have 23 more of those bad boys. So if we think about that 58 million base pair molecule of DNA, your chromosome number 19, what it's hanging out as in most of your cells most of the time is that sort of ball of spaghetti action, right? In with all of your other chromosomes because it's in the structure of chromatin. Whenever scientists were first studying, trying to figure out even what the genetic material was, cell division was one of the places where key information came from, because researchers were able to actually see individual pieces of DNA. And the way that this happens is during mitosis, of course, during cellular division. And so if early researchers, and even now, wanted to study individual pieces of DNA or even figure out, like, where does this gene map, what they needed to do was to create something called a karyotype. And so what they did was they would take a cell or many cells that were undergoing cell division, undergoing mitosis. So indicated by this image here. And typically it was during metaphase of mitosis when the chromosomes were all nicely condensed, and then they could be separated. So then, only then, could you figure out what piece of DNA goes with what, right? Oops. Sorry, made that do something weird. And then this is how we get karyotypes. This is how we get those images that you're probably familiar with whenever people talk about DNA, when people talk about chromosomes, they talk about karyotypes. And so what happens to make a karyotype is that cells are taken during cellular division, and each of those pieces of DNA is separated out, and then they're sorted into these karyotypes. So why is it then, cool, got it, karyotypes, I'm good with that. Why the heck do karyotypes sometimes look like this, but other times look like that? So maybe you didn't at first really kind of notice the difference, but let's take a closer look at this. So if we look at this karyotype, the image is on both sides. The image on the left side, which I'm going to circle in white, that's chromosome 1, human chromosome 1. The image on the right, which I'm going to separate in green, that is also human chromosome 1. The area encased in a blue rectangle labeled I, that's one copy of chromosome 1. I mean labeled A. Did I say I? Labeled A. This one right here. That's one copy of chromosome 1. And then the one labeled B is another copy of chromosome 1. So then why is it that in the left image you just kind of have one little crinkly caterpillar in each box, and on the other side you have two crinkly caterpillars in each box? So getting into that, we're going to be talking about ploidy, haploid and diploid, to help answer this question so that it's not confusing when you're looking at these images. So that's next.